Hello students, today we will start about a new topic, that is cytoplasmic inheritance. What is meant by cytoplasmic inheritance? The inheritance of characters that are controlled by some of the genes that are located in the cytoplasm. And there is some name for such type of genes that is called as cytogenes. Now, if you see the sentence framing or definition of that cytoplasmic inheritance, inheritance of the characters that are controlled by cytogenes of cytoplasm. This cytoplasmic inheritance is discovered and given by a person, that is Corens. Actually, this is one of the MCQ, and the cytogenes and plasmon terms are also MCQs I have seen in some of the practice books. Next, if we come to know what is meant by these cytogenes, these are located in the cytoplasm, and they are called as cytogenes, also called as plasma genes, also called as extranuclear genes. Now, in a cell, in the total content of the cytoplasm, how much content of cytogenes are present, or how many number of cytogenes are present, that total content combinedly is called as plasmon. The total content of cytogenes in the cytoplasm of one cell is called as plasmon. Then what about the name of genes that are present inside the nucleus? The total content of genes inside the nucleus are called as karyogene. Karyon means nucleus. Now, the inheritance of these cytogenes occurs by the female gametes or the female organism, because egg is having sufficient content of cytoplasm and cytogenes will be existed inside that. But in case of male gametes like sperms, there is very much negligible content of cytoplasm. Sometimes that may also considered as to be absent. So we do not consider the transportation of such type of cytoplasmic inheritance through the sperms or male gametes. Cytoplasmic inheritance by chloroplast, because chloroplast is one of the organelle that is present in the cytoplasm, is seen in plant called 4 o'clock plant. 4 o'clock plant. Now, 4 o'clock plant is also called as Mirabilis jalapa. Cytoplasmic inheritance with the help of mitochondria. It is seen in case of Gia maize plant. Now, this is discovered and given by Rodi. Okay. This person has given the identification that the genetic material, that is double-stranded, circular, naked DNA that is present in the mitochondria, is responsible for cytoplasmic inheritance, means transferring of whatever the characters are there with the help of cytogenes. Now, here, chloroplast and mitochondria are the two cell organelles in the eukaryotic cell that have double-stranded, circular, naked DNA. And so, these both are called as autonomous organelles. Means, they have their own genetic information. If any repair or any further characteristic features are necessary for those two types of organelles, they can do by their own. Now, what are the other examples to study cytoplasmic inheritance? One example is, other examples are shell coiling, on snails. This is one example. Second example is kappa particles of Paramecium aurelia. So let's see about these two types of examples also. But one is green colored branch, another one is pale green colored branch, third one is variated. Variated means different colored branches. Variated means some different types. So this branch has chloroplast, which is green in color. This pale green branch has leucoplast, which is colorless or white in color. Variated, this type of branch has chloroplast, leucoplast, and chromoplast. Now, what I said is, if a flower is produced from this branch and that flower's female gynoecium is there, if that is crossed with the pollen of this flower's pollen, but it don't know that it is crossing with that another type of branched pollen, then it will give out some different color. It can give either, since here female structure is from green color, it will produce only green colored branch again. Because we know that female gametes are having only cytoplasmic inheritance and pollen is not responsible for cytoplasmic inheritance. If any flower that produces egg cell and if that egg is crossed or fused with male gamete of unknown branch or unknown flower, then the obtained new offspring will be of will depend on egg cytoplasm. Egg cytoplasm. This is what I have said just before. See, here, if when we have taken the egg from 3 different branches and we crossed with again another 3 different types of pollen grains from 3 different branches, then the obtained offspring in the next generation, it depends upon the characteristic features that are inherited from the egg. So when this variated flower or gynoecium from this variated branch, even it is fused with the pollen of green colored branched flower, then obtained will be of variated type only. In the same way of this pale green also. Pale green branch is producing the egg. It is crossed with the pollen grain of some other different types of branches. Still the obtained offsprings will be of pale green. The same with green type of branch also. Here we can see whatever the characters that are exhibited in the offsprings are depending on the egg cytoplasm characteristic features and also the cytogenes only. Next, so example 1, shell coiling in snail. We see 2 different types of shell coilings. One is a right-handed helix that is clockwise directed and a left-handed helix that is anti-clockwise coiled type of shell. So the scientific name of this one or zoological name of this one is Limnaca. Yes. Perigra. Limnaca perigra. This is the scientific name or zoological name of the snails. The shell of snail contains a protein that is coded by the cytogenes. So, there are two types of shell shapes. So, one is called as dextral shape. Another is called as sinistral shape. Now, what is meant by dextral shape? It is also called as right-handed helix or clockwise directed shell. Also called as right-handed coil or clockwise directed. Now, this dextral shape is considered to be due to only the dominant genes. Dominant genes, again, it means a gene exists in the form of alleles, and alleles are always in the contrasting pairs, means two different types of characteristic features are exhibited by the one type of allele when they are having another type of alleles, like for suppose the dominant alleles are there, dominant type are there, when another contrasting type of their own, another type of allele forms are there, only this type are exhibited and these get suppressed, they are not exhibited. Then we are calling such type of genes as dominant genes. Now, what about this one? These, when they are suppressed in another allele pair presence, then they are called as recessive forms. So here, right-handed helix or clockwise directed shell of snail is considered to be due to dominant alleles. Now, sinistral shape. This is also called as left-handed helix or anti-clockwise directed. Left-handed coil form or anti-clockwise directed shape. Now, it is considered to be due to recessive alleles. And the snail that is having such a type of recessive alleles will not produce the protein that is responsible for right-handed helix form. So it has given anti-clockwise directed shell coil. Next, we will see the last example, that is kappa particles of Paramecium aurelia. So, what are kappa particles? Kappa particles are also called as kappa organisms. These are considered to be gram-negative symbiotic bacteria that are present only in the cytoplasm of some strains of Paramecium. Then we call such type of Paramecium as killer strains present in cytoplasm of some paramecium aurelia. Now, when such type of paramecium aurelia, along with these kappa organisms or the symbiotic bacteria, that is gram-negative symbiotic bacteria, is present in their cytoplasm, and if such type of strains are released into water or whatever the medium that they are residing, then they produce a special type of toxin called as paramecin. In two different forms. What is first form? So, paramecium aurelia, this is the protozoan with the kappa particles. Is called killer strain. Second type, paramecium aurelia or any other species without kappa particles is called normal or sensitive strain. Now, when this killer strain is released into the medium that it is residing, it produces a special type of toxin called as paramecin. Killer strain in medium releases toxin called paramecin. So, what is the nature of this paramecin? It kills all types of or other types of strains other than the means killer strains. Whatever are there, paramecium varieties, those are killed by this type of toxin called as paramecin. So, it kills other paramecium strains. Now, these kappa particles are discovered by Sonneborn. It is discovered by Sonneborn. Now, there is very important factor that we have to study about these killer strains, that this mode of transportation of kappa particles from killer strains to other types of strains and converting again them back to killer strains should be done within 3 minutes of conjugation. We know that the sexual reproduction of exchange of nucleus and the cytoplasm happens in case of paramecium only by the process of conjugation process. The sexual reproduction is called, in paramecium is called conjugation process. During this process, the exchange of cytoplasm and also the nuclear material happens, and during that time, sometimes the toxin is entering into other type of paramecium and killing the other strains. It is okay. At the same time, sometimes, very rarely, normal strains are also converted into kappa strains, means or the killer strains. Now, that conjugation should happen within 3 minutes of a time period. So, this cytoplasmic inheritance should happen within less than 3 minutes. If not, the correct process of cytoplasmic inheritance may not happen, and the sensitive strain may be toxically prone to this paramecin type and can be killed. So this is about kappa particles. Next we will see about new topic, that is chromosomal theory of inheritance. Chromosomal theory of inheritance is given by Sutton and Boveri in the year 1902. Before that, microscope was not discovered, so that the studies regarding such type of chromosomal inheritance and all that is not that much advanced. So microscopes are discovered in 1902. Then only the studies regarding the chromosomal movement during anaphase of meiosis was done. Sutton and Boveri, they used this knowledge of chromosomal movement to explain Mendel's second law, that is law of segregation. We also know that law of segregation is universally applicable. And they concluded that this pairing and segregation of chromosomes led to the segregation of genes or factors. Factors is the term that is used by Mendel. Mendel also used the term called as element to represent the genes and that are carried by chromosomes. So these are already existed inside the DNA particles, which we call as genes now. Among both the scientists, Sutton and Boveri, Sutton has united these Mendel's laws with that of segregating chromosomes during the anaphase, and he has united these both concepts and gave us chromosomal theory of inheritance. Now, chromosomal theory of inheritance is represented in diagrammatic form. We will see how one complete diploid cell which is having two different types of non-allelic chromosomes. We can also consider this one as homologous chromosomes also. Means here one set of chromosomes which are uniting, and these are another set of chromosomes which are uniting, but these both are being considered as homologous chromosomes separately, and they are segregated into the offsprings or the germ cells that are produced in the end. So we can conclude this first diagram as this is diploid with 2n condition and is a cell, means one complete cell, a cell that later forms haploid germ cells. Now, these both are considered as homologous, these both are considered as homologous chromosomes. This we can consider as the G1 stage of the interphase. During the G1 phase, chromosomes are arranged. Means these two chromosomes are responsible for one type of character. These chromosomes can be responsible for another type of character. Suppose think that these are arranged in the G1 stage like this. Coming to the next stage, what happens in the G2 phase? The chromosomes which are existed like this, they will be converted after G2 and next in zygotine stage. Zygotine is one of the step or stage in prophase 1 of meiosis. So here the chromosomes are modified into bivalents, into bivalent forms. So the bivalent form of chromosomes is seen in the next second diagram here. So what are bivalent chromosomes? Bivalent is a means stage of the chromosomes in which the homologous chromosomes are paired. For suppose think these two are side by side like this. Now, if they are arranged like this, then there will be crossing over in the next stage, that is packeting. But here I did not represent the crossing over because of lack of space here. But bivalent chromosomes are those which are ready to pair with each other. In the next stage, packeting, they will cross and they will show the variations. So here, since two chromosomes are pairing, we call them as homologous chromosomes, and there are two number of chromosomes here that are going to pair. So we are telling bi. Bi means two different chromosomes or homologous chromosomes that are going to cross with each other in the next step. But at the same time, how many chromatids are there? 1, 2, 3, 4. 4 chromatids are there. So we can also give a term like this. At this stage, they also exhibit the tetrad form. Tetrad means 4 chromatids will be seen during the crossing over. crossing over happens in pachytene. Now, here I did not show the crossing over, so I have written just the zygote term. Since two homologous chromosomes are going to pair, I have used the term like bivalent. It is the stage at which synapses will occur. Means how many number of centromeres are there in one set of homologous chromosomes? In case of bivalents which are pairing, there will be 2 number of centromeres, 2 number of chromosomes, 4 number of tetrades. Tetrade is the term that we use for the chromatids that are present. So here we are seeing 2 sets of chromosomes which are responsible for 2 different types of characters. Now, this one we can say after zygote, it is going to the next step, that is anaphase. Anaphase 1. During the anaphase, the chromosomes are moved to opposite poles. So this can be considered as the pole where centrioles exist. This also can be considered as the poles. Means during anaphase, there is no splitting of centromere. Chromosomes will be moved to opposite poles as it is. Since we have taken 2 different chromosomes responsible for 2 different types of characteristic features, chromosomes are moved as it is to the opposite poles. Next, in telophase 1, there will be division of the cell into 2 different baby cells or the offsprings. This is just the finishing of meiosis 1 only. So after telophase, we are getting 2 different cells that have attained 2 different types of chromosomes according to this diagrammatic representation. Now, after that, again, these have to enter into meiosis 2. So, after telophase 1 and obtaining of 2 cells with 2 different sets of chromosomes, by this we are telling each cell is haploid in condition because the number of chromosomes have become to half in each baby cell. Now entering into meiosis 2, and the diagrammatic representation which I have shown here indicates anaphase 2. Anaphase 2. After in anaphase, what is happening? The anaphase 2, what is happening? The splitting of chromosomes. This one, here. The chromosomes is splitted according to the separation of chromatids separately vertically, and these are moved to again opposite poles. The spindle fibers will be dragging each the chromatid part towards the opposite poles. So this cell is represented like this and this cell is represented in this form. Now, when meiosis 2 is ending, then the stage will be telophase 2. Telophase 2. In the telophase 2, each cell shows a division that is equal to mitosis. So we are getting again the same number of haploid number of cells. Meiosis 2 is equal to mitosis. Again I am telling at anaphase 1 there is no centromere splitting, but at anaphase 2 there is splitting of the centromeres and we are getting same haploid type of germ cells at the end. This total diagrammatic representation is according to your NCERT page number 82 page diagrammatic representation. So please go through these points. Whatever I have written the extra points here. We can also use a term like the type of chromosomes that are involving for the production of germ cells are nothing but considered as non-allelic chromosomes.